Hi guys, welcome to a quick revision session on the gene technology, and exactly what we will be looking at in this video is how to make fragments, copies of the fragments of DNA. So we will follow 5 steps here, from isolation through insertion, transformation, identification and the growth. So we're starting with the isolation. Isolation is a process where you want to obtain a sequence of the gene, so that's a section of the DNA coding for the protein of your choice. So there are 3 ways of doing this. We could use the enzyme reverse transcriptase, restriction endonuclease enzyme, or the gene machine. We will start with the reverse transcriptase. You did learn about reverse transcriptase with the topic of immunity with the replication of HIV. With the replication of HIV. So, reverse transcriptase is an enzyme that uses mRNA as a template to make DNA. mRNA is a single-stranded nucleic acid. So I'm going to put some bases here. So reverse transcriptase has the ability to use mRNA as a template to make complementary cDNA. This complementary cDNA, for time being now, it's going to be just single-stranded. So A, it's going to go adenine with thymine, uracil with adenine, guanine with cytosine, cytosine with guanine, adenine with thymine, and uracil with adenine. This reverse transcriptase enzyme allowed us, so that was thanks to reverse transcriptase enzyme, this enzyme allowed us to use mRNA, a single-stranded nucleic acid, as a template to make a complementary, so cDNA. This cDNA, it's single-stranded. And now we, of course, want to make it into double-stranded DNA. So as we can predict here, we will need help of another enzyme. So thymine will go with adenine, adenine with thymine, cytosine with guanine, guanine with cytosine, thymine with adenine, and adenine with thymine. Of course, the enzyme here that will allow for this double-stranded DNA to be achieved. So there we go, our double-stranded DNA. It's, of course, DNA polymerase. The advantage of using reverse transcriptase against restriction nuclease, which we will look at in a second, it's the fact that within those sequences here, we have no introns. So remember, introns are non-coding sequences on DNA. Why we don't have them? Because we've started with mRNA. mRNA has been formed previously as the process of transcription, and after transcription we had splicing, which was the removal of introns. So this is the action of reverse transcriptase, which allows us to get the section of the gene of our interest. Other way of getting the gene of our interest is using restriction endonuclease. This enzyme has the ability to cut DNA in the position that we are interested at. So our DNA could be cut in 2 ways. So imagine this is our DNA. Okay, I can cut it straight like this. So after the process of cutting, I'm going to get straight ends. No bases will be sticking out, and those ends are called blunt ends. So imagine this is how I chop my DNA. Or I could cut that DNA, which is the more common process, at different positions here, not straight, and at the end I'm achieving the section of DNA like this. So as we can see, we have here exposed bases. So those are exposed bases, those are exposed bases. And that way of cutting DNA gives us the product which we would call sticky ends. Sticky ends are really important for the next parts of our processes when we will be looking at insertion and so on. The way we cut those fragments of DNA, it's actually located at the specific sequences. And the sequences where we cut to produce sticky ends are called palindromic sequence. And here, obviously, we do have introns as we've started with the original fragments of DNA. Final method, it's the method of using the computer-based program called Gene Machine. So let me use the terminology as artificial nucleotides, so the computer-based nucleotides that we are going to feed into the computer to give us the sequence of that specific gene. Those nucleotides are called oligonucleotides. Here the advantage is as well that we have no introns, because we will feed into the computer just the sequence of the nucleotide sequence of the bases that codes for the specific protein of our interest. So that was the first process of isolation. We can achieve the sequence of the gene coding for the specific protein using those three following methods. What will then happen with that gene that we have achieved? So color in green again, I'll go for the green color. That gene is going to then be inserted into the plasmid. So that's the next step. We've got insertion. Insertion of that gene of our interest, so remember it was the green, into the vector. Vectors are plasmids, and they have the ability to carry DNA fragments of that gene that we've achieved in the first process in the isolation. So you should remember that plasmids are single loops of DNA, and plasmids got the function to carry information for the antibiotic resistance. They carry many of the genes for this antibiotic resistance. So imagine I'm starting with my plasmid, which has a gene A, coding for antibiotic A, any antibiotic of your choice. For instance, amoxicillin. And this is, I'm going to color that part of the plasmid blue, codes for gene A. Also, I'm going to have a gene B. And let's say this gene, it's coding for the antibiotic resistance, this time to penicillin. Okay, so I've got plasmid with at least two genes coding for the resistance to different antibiotics. So, as we've said, we want to now insert our gene, the green one that we've achieved on the previous slide, into the vector, into the plasmid. So what I need to do here, I need to cut open the plasmid. And our gene that we've achieved in the previous stage, obviously, remember, has sticky ends. So those are the sticky ends that I've achieved in the isolation stage, and want to put them now into plasmid. So logically thinking, I need to make sure that the part I will cut of that plasmid is complementary to my sticky end, so I can easily replace the parts. So I'm going to have to use exactly the same restriction endonuclease, because again, due to specific 3D tertiary structure, that enzyme will allow me to cut exactly complementary section. So cut open plasmid using the same restriction endonuclease. So I'm going to target gene B. I'm going to cut at gene B. So at the same sequence, so it's complementary. So now my modified recombinant plasmid will still carry the gene A, telling me about the antibiotic resistance to amoxicillin. However, this gene B now will be replaced with my sticky ends. So I'm putting the sticky ends here. Okay, so that is the gene coding for my specific protein. Still, plasmid carries the gene A information. So technically saying, this plasmid, now it's still carrying the antibiotic resistance to the antibiotic A, however lost the resistance to antibiotic B because I've removed that gene and I've replaced that with the sticky ends. And of course I need to join those fragments together, and I'm going to use the enzyme to join them together. And the enzyme used to insert and stick in the sticky ends with the plasmid, it's called DNA ligase. So DNA ligase, it's going to allow me to join those two fragments of DNA, so my sticky ends and the plasmid ends together. Okay, and that was the process of insertion. So remember the key point here, I've lost the resistance to antibiotic B by doing that. So the vector, the plasmid, will now carry DNA to our next stage, which is transformation. So we want to insert the recombinant plasmid into the host cell. And the host cell that I'm going to use is the bacteria cell. So remember, still got the antibiotic resistance A gene, and I've got the sticky ends. However, the process of insertion into bacteria cell, into the host cell, is not going to be that easy, because for the simple fact, I've got here many layers. I've got cell membrane, and I've got cell wall, which is the main problem here. And I've got capsule. So how do I overcome the biggest problem, which is the cell wall? Well, I need to make the cell wall more permeable to allow my plasmid to be injected. Or I could say transfer with the pipette. So how am I going to do it? How the membrane can become, cell wall become more permeable? So there are two processes that need to take place. Process number one, I need to grow this plasmid and bacteria on medium containing calcium ions. And the final process, I need to increase temperature. Those two processes will make the cell wall more permeable so I can inject my plasmid. So obviously, as in all of the processes, scientific mechanisms, I need to now check if actually this bacteria, my host cell, has taken up the plasmid. So that leads us to the next process, which is identification. So now I'm going to identify if my plasmid has been taken. So remember, my plasmid should still contain the antibiotic resistance A and should contain my sticky ends. And just to remind you, my original plasmid was slightly different, so I'll talk about it now. So one of the ways of checking if the bacteria has taken up the plasmids, it's using gene markers. We've got three of them. First one, we're going to talk about in a second. We're going to go for the easy ones, straightforward ones. So I could use fluorescent proteins from jellyfish. That if I use them, the bacteria that has taken up the plasmid will just glow in the dark. Or I could use the enzyme markers, and if bacteria has taken up the plasmids, the bacteria won't be able to make the enzyme lactase. So the substrate I'm using here will be still colorless. However, if the bacteria did not take up the plasmid, they can still make lactase, and the presence of the substrate will change the color here into blue with the lactase. With the fact that they cannot make lactase, of course, enzyme and substrate do not interact together. Hence, the solution will be still colorless. And let's go for the the oldest method, but the most common on the exam now, which is replica plating. That uses the antibiotic resistance. So how this is done? Remember, my original plasmid has carried 2 genes before the insertion of my sticky ends. I had gene A and had gene B. Okay, so we had gene A and we had the gene B. My recombinant plasmid after insertion of the sticky ends now carries still gene A, but doesn't carry the gene B, it carries my sticky ends instead. So the sequence that will code for protein of my interest. So how easily do I check if the bacteria has taken up the plasmid? Well, really easily. So if gene A, let's say, allows them to be, carries the antibiotic resistance to let's go for the, I don't know what we could go for, amoxicillin. Both of those plasmids in the bacteria, so bacteria will survive if I will treat them with amoxicillin. And let's imagine that gene B will give the resistance to the penicillin. So my original plasmid will allow this bacteria to survive with the penicillin. However, my recombinant plasmid now will not, because it doesn't carry that gene anymore, it carries my sticky ends. So technically saying, all the bacteria now will die. And that's how easily I can recognize if the plasmid has been taken up by the host cell. So now I've done all the checks. I can see that my gene was taken up, so I'm ready for the growth. So I can make many copies of my gene. And this process, it's called PCR, polymerase chain reaction. So I'm going to make copies of my gene. However, it's different than DNA replication because it happens outside of the living organisms. Hence, it's called in vitro. This process is made of 3 main stages. First stage is the separation of DNA. Here, we will not use DNA helicase as we've used in DNA replication. Instead, we will use the high temperature of 95 degrees. This high temperature of 95 degrees will allow those strands to separate. So we've got 2 separated strands of DNA. Next stage is the annealing of primers. For this, I need to decrease temperature to around 55 degrees. What are the primers? primers are the single-stranded short pieces, fragments of DNA, which we will add at the opposite ends. So here and there. The primers got two main jobs. Job number one is to label the beginning of the synthesis, and job number two is to prevent those strands from rejoining, so they don't rejoin, and the synthesis then can follow on both strands. So finally, synthesis of DNA happens at the temperature of 72 degrees, which is the optimum temperature for DNA polymerase, which of course joins nucleotides. So as the effect, I'm going to achieve, obviously, double amount of DNA, but in exactly different way to what you've learned in DNA replication, because remember that process happens outside of the living organisms. It's called in vitro. So that's the overview of the large section of the gene technology, when we've made the fragments of our genes, of our insertions, following isolation, insertion, transformation, identification, and the growth. Make sure now you obviously get confident with the content, know your model answers, know all of the new words that we've mentioned about